Okay, so we are going to go over the prenatal and early childhood programming of adult disease. At the end of this section, you should be able to describe the biological mechanisms by which prenatal and early childhood environments impact adult health outcomes. We have already discussed some of these biological mechanisms, but today we're going to go into a little bit more detail. Today, it's all about epigenetics. I want you to be able to define toxic stress and identify early life as a critical window of vulnerability, and discuss how the science of epigenetics influences our understanding of disease etiology and our approach to developing new interventions. And so today we are really going to build on that life course health development model. But first, review. So let's go over the HPA axis. You have probably gone over this before multiple times. Hopefully you have. Hopefully you have it memorized, but start with stress, right? It's usually your amygdala that senses stress and activates this response system. And then it sends a signal to your hypothalamus to release CRH, which sends a signal only a short distance away to your pituitary gland, which releases ACTH. And then that sends a signal to your adrenals to release glucocorticoids and catecholamines. And remember, this signal has different effects depending on the type of receptor that it binds to and depending on the type of cell that it binds to, right? And so you have effects in your immune system, which we haven't talked about yet, but we will. Basically your muscle, liver, fat, all to mobilize those stores of energy, right? Because if you're trying to survive, you need to be able to fuel your getaway, and then to your cardiovascular system. And then you have this negative feedback system that once your adrenals release catecholamine or glucocorticoids, which is cortisol, it sends a negative signal to the hypothalamus and to your pituitary gland to shut it down. But it's a little bit more complex than that. And so let's go through it in a different way. So here's your hypothalamus, releases CRH, tells your pituitary to release ACTH, tells your adrenal glands to release cortisol. And then that has a negative feedback both to the hypothalamus and to the pituitary, but it's a weaker negative feedback. The actual feedback involves the hippocampus. And remember, we talked about hippocampus as being, the place in your brain that makes memories. It's intimately involved in memory formation. But what happens is cortisol binds to glucocorticoid receptors in your hippocampus. And then the hippocampus shuts off the HPA axis. So a simplified version is the amygdala triggers your HPA axis. The hippocampus shuts it down. And cortisol has a negative feedback both to the HPA axis and a positive feedback to the hippocampus. So this part is a little bit confusing. What I want you to know is that the hippocampus has receptors for cortisol. They have it is full of glucocorticoid receptors. And so as it binds to those, then the hippocampus now has this enough signal to shut down this whole stress response. And this is going to be very important moving forward. So biological mechanisms for disease. Remember that environmental exposures can impact the genome and epigenome, right? And so either way, whether we're talking about mutations that affect gene expression, right? But most mutations don't, or whether we're talking about epigenetic changes that affect gene expression, both can result in a particular phenotype, right? And so and both can control gene expression. So epigenetics, the function is to control gene expression by turning genes on or off in response to environmental signals. The mechanisms, remember, we have DNA methylation, we have histone modification, and we also have the RNA I. The modifications here can be malleable, which means that they can change, and they can be inherited. And that's something we didn't spend a lot of time talking about the last time, but that's what this section is all about. This image should just be review. It was in the genetic section. And then the epigenome changes in response to environmental signals. And a lot of these images are from the Utah website. Please spend time reviewing that website so you have a better understanding of these mechanisms. But remember, signals can come from everywhere. It could be like the cell right next to it. It could be even like within the cell. It can be hormones from like across the body. And it can be environmental signals as well. And there's no like hierarchy of, importance. They all play a role in shaping the epigenome. So the epigenome also is a form of cellular memory. Okay. The epigenome allows cells to remember their past experiences long after the signals fade. And this is one way that, cells differentiate. And we didn't really talk about this within this, framework and framing it like this. But when you have cell division, right, like you're an embryo, and then first you're like one cell and then two and then four, right? Initially, there are genes that get shut off or epigenetically silenced, right? That makes sense. But then after they divide, if it's not a genetic mutation, how will the daughter cells know what to do unless there is some kind of inheritance to epigenetic mechanisms, right? So think about that for a minute, because you understand differentiation, but there has to be some form of cellular memory if additional divisions all still track along the same process. So it becomes a neuron and a specialized neuron. Well, it has to have some kind of, tag or something that shuts those genes down in future generations. And that's what we see. So it's not necessarily new, it's just a different way of looking at it or understanding it. So reprogramming and imprinting are mechanisms that are important in understanding epigenetics. So reprogramming erases most of the epigenetic tags so that the fertilized egg can develop into any type of cell. So, at some point you have to like shed all of those silencing mechanisms so you can become whatever, right? And that's usually after conception or, fertilization, I should say. So those tags have to be lost. But you also have imprinting, which makes things a little bit more interesting, right? Because there's some way that our cells know that certain genes come from our, male genetic donor, and certain genes come from the female genetic donor. And with imprinting, they have to have some form of memory, right? To be able to remember that. And so about 1% of genes escape this reprogramming, and that's where we get the imprinting. So there are certain windows of vulnerability, and we've talked about this concept before, right? We're going to focus first on preconception and the prenatal period. So In the prenatal period, what's important to understand is that there are actually 3 generations that are directly exposed to the same environmental conditions at the same time. The mother is being exposed, right? The fetus is being exposed, and particularly for females, when you are a fetus and a developing embryo, you have all of the eggs you will ever have for your entire life. And so, if a particular environmental signal either causes a mutation in the genome or an epigenetic silencing in the genome, it could potentially affect 3 generations. And so this is good to know about because it's a little bit difficult to study this effect because you do have to look at generations. And so this might be one reason why it's taken us a while to actually figure this whole process out. So nutrition in the epigenome, you have seen this diagram before. Major points to take away is that, your food supplies your body with like essential nutrients, and some of those nutrients are used in epigenetics, right? You don't necessarily have to memorize all of these. The major one is folic acid or folate, right? And so, how are we going to make this relevant? Well, this is relevant because the diet during early development can have long-lasting effects. So if you have this epigenetic silencing that's supposed to happen or not, you need the right building blocks to be able to do this. And so if you are missing some of these building blocks early on, especially during embryonic development, then these can have horrible effects. So the first clip that we are going to watch is of the animal models. And you can access the link to this clip through the slides. You can also access it through the playlist link, or you could just watch the whole movie through. But for those of you who don't want to watch the entire movie, I'll go ahead and share the link with you or the clip with you.
Some of the first clues that our lived experience could alter the way our genes work came from a set of experiments involving some genetically identical mice that somehow weren't identical.
The brown pseudo-agouti mouse weighs around 27 grams. Here is the yellow mouse that weighs around 52 grams.
How could it be that some mice are brown and thin, others yellow and obese, when they all shared the exact same DNA? It's what their mothers were fed while they were pregnant. Some were fed pellets rich in methyl groups, a molecule commonly found in fruits and vegetables. Those mice were more likely to give birth to brown, skinny pups. But mothers fed a diet with fewer methyl groups were more likely to give birth to pups that were yellow and obese.
Whoops, tipped the scale.
These studies were first conducted at Duke University under biologist Randy Girdle. Jurdel's team, including Dana Dolinoy, zeroed in on one particular gene in search of an explanation. The agouti gene codes for yellow fur. Curiously, it's also related to obesity. Jurdel's team found that the methyl groups fed to the pregnant mothers passed through to the developing pups and attached to their agouti genes. It's a process called methylation.
DNA methylation is the addition of a single carbon in the form of a methyl group. So this is a carbon and 3 hydrogens that sits down on DNA and shuts genes off.
With the gene on or active, more mice were born yellow and grew obese. With the gene silenced, more mice were born brown and remained thin. Same genes, very different results. The difference is whether particular genes are switched on or off, expressed or silenced. The genes we inherit are fixed for life. But there's an entire network of molecules that influences which genes are expressed by making them active or shutting them down. Like a volume control or a dimmer switch, it's called the epigenome.
Well, literally, it just means in addition to or above the genome.
The epigenome, it's a system of molecular switches that interact